my name is Nicholas Kovac. My advisor is Dr. Lauren Williams, and we are at Georgia Tech in Atlanta, Georgia. I'm a bioinformatics PhD candidate in the School of Chemistry and Biochemistry, and my talk today is titled Frozen in Time, the History of Proteins. So I'll be talking about a really large molecular structure, but I swear it pertains to the origin of life, so just bear with the introduction. I'll get to it eventually. So just a brief introduction about the central dogma of molecular biology. I'm sure all of you guys are familiar with this, but just to touch base with it for some of those geologists or some other people. So here on the left we have DNA, and it's a nucleic acid polymer, and on it we have encoded genes, amongst other things, but genes are what we can talk about. And over here in the blue we have an enzyme, RNA polymerase, which reads the DNA and looks at just the genes and copies those into RNA, another nucleic acid polymer. And so this RNA then just has just the one gene on it, and this gene on the mRNA goes to the ribosome. And the ribosome then reads the mRNA and makes a protein, an amino acid polymer. And so I will be talking about the ribosome right here, the key, and it, which reads RNA and makes protein. So, looking at the ribosome, it has two subunits, the small subunit and the large subunit. The small subunit is on the left, and the large subunit is on the right. RNA I have colored in pink and yellow, and in blue are the ribosomal proteins. And then right there in the green, at the very center of the large subunit, is where peptide bond catalysis happens. That is called the peptidyl-transferase center. And this peptidyl-transferase center is exclusively RNA. So the ribosome is a ribozyme. Okay, so like I said, the ribosome reads RNA and it makes protein, or it reads nucleic acid polymers and it makes an amino acid polymer, or another way of saying that is its input is information and its output is function. So in 2009, the Nobel Prize in Chemistry was awarded to Venki Ramakrishnan, Thomas Steitz, and Adi Yonath for the studies of the structure and function of the ribosome. And they made this really cool animation. And it's very complex, but this is the introduction of the ribosome, so I just want to show you it. On the bottom here, we have the small subunit that's in transparent pink. On the top, we have in this transparent yellow, that would be the large subunit. The mRNA is in blue, and it's being read by the small subunit. And these structures going in and out of here are tRNAs, and they're bringing amino acids. And these amino acids are brought to the peptidyl-transferase center, right in the middle of the large subunit, and they get strung together to make a peptide that then emerges out the back of the ribosome. So now to look at the sequence of the ribosome. So I'm sure many of you are familiar with phylogenetic trees of life, where we have the three domains of life: bacteria, archaea, and eukarya. Over here we have animals, and they're distantly related to fungi and plants. And then we have archaeans, which are then even farther distantly related to eukaryotes. And then bacteria are all over here. And so these phylogenetic trees are made by looking at the sequence of ribosomal genes. So the ribosomal RNA and ribosomal proteins are encoded on the genome of every organism. And so just the ribosomal RNA genes are just not made into protein. They just stay as RNA, but we can just be looking at the sequence, and that's how we construct these phylogenetic trees. So the ribosome has within it like the interrelatedness of all of life. So looking at the ribosome from across the tree of life, we can see that bacteria, the large subunit, has 2 ribosomal RNAs and 33 ribosomal proteins, whereas the small subunit has 1 ribosomal RNA and 22 ribosomal proteins. Archaeans then have a few more ribosomal proteins in both its large subunit and small subunit. And then eukaryotes have one or two more ribosomal RNAs in its large subunit and a few more ribosomal proteins in both its LSU, large subunit, and SSU, small subunit. And so bacteria's ribosomes are pretty small, whereas archaeans are a little bit bigger, and eukaryotes have very large ribosomes. So to conclude this introduction on the ribosome, its sequence and structure, ribosomes are made of both RNA and protein, ribosomal RNA and protein sequences describe the interrelatedness of life, and ribosome size is highly correlated with species complexity. Here's another phylogenetic tree. So on the top we have Homo sapiens, and below that shrimps. So those are mammals, and then farther down we have chicken and let's see, mosquito right there. So those are animals. And then farther down here we have single-cellular eukaryotes like Strepsirrhiniaceae, fungus, and I don't know what else we have here. I think Plasmodium falciparum, which is the parasite that causes malaria. And then down here I just have archaea and bacteria, because what I'm trying to show in this phylogenetic tree is the size of the ribosome. So bacteria, their ribosome is all about the same size, but the sequence is very different. And the same with archaeans. Their ribosome is a little bit larger than bacteria, but their sequence is very different within the whole entire domain. Whereas eukaryotes, there's like a really big difference in their size of the ribosomes. And so that's what I have over here in these circles. So bacteria, the ribosome is small, and archaeans is also fairly small. But then these single-cellular eukaryotes have, medium-sized ribosomes. These animals have even larger ribosomes, and then mammals have the largest ribosomes that we see. And so this is very highly correlated to how complex the species is. People used to think, oh, well, maybe like the length of the DNA of an organism would tell you how complex that species is, but there's some really big anomalies that really just throw it off. For instance, the marbled lungfish its genome was 120 billion nucleotides long, whereas humans, it's 6 billion. So clearly that's not like a really good indication of how complex the species is. Okay, so what am I doing in my project? So I'm looking at the ribosome, and I'm constructing a data set of its structure. And the reason I'm doing that is because the ribosome is the origin of life, and it contains within its structure and sequence the history of protein folding. And I believe that by mining the structure of it and the sequence, that I can unravel how life has evolved. So, like I said, I'm looking at structures of ribosomes, and in 2009, the Nobel Prize was awarded for the structure for crystallizing the ribosome. And since then, we have quite a few structures. So we have bacteria, E. coli, and Thermus thermophilus. On the left, we have the large subunit, and on the right, we have the small subunit. We have the archaeans, Haloarchaea marsimorti, and Pyrococcus furiosus. Haloarchaea, we only have the large subunit, not the small subunit. We couldn't get it crystallized. We also have the protozoan, a single-cellular eukaryote. Again, we only have the large subunit. We have the single-cellular fungi, Saccharomyces cerevisiae. The animals, Drosophila melanogaster and Homo sapiens, fruit fly and humans. And we also have the human protozoan parasites that cause malaria and African sleeping sickness. And so what happens when we align all these onto each other? Well, as you can see, bacteria and archaea have pretty small ribosomes. But then when you start putting on the eukaryotes, it gets a little bigger and larger and larger. And then I have these human protozoan parasites over here for a discussion later on that I can maybe have. But for the focus of this talk, I'm not going to talk too much about them. But as you can see, these structures are quite a bit larger than the E. coli and other bacteria species. And what we get is we get this big glob of like hundreds of millions of atoms. And we really just can't make sense of it when you look at that. But when you look deep within its structure, you can see that the structures of the RNA and the protein that make up the ribosome in the center of it are miraculously just similar. Their sequence is different, but their structure is just crazy the same, going from the center all the way out to near the surface of it. It's only the surface of the ribosomal structures that we see any variation. So the center of it is just a common core of structure that just doesn't divulge from any species that we know of. And it's when we look at the surface then we can see how this RNA has evolved. So looking at that one helix from the last slide, this is helix 25. And I'll just use this as an example to show you how ribosomal RNA has evolved. So it's helix 25 of bacteria. We have archaeans, protozoans, fungi, animals, and our parasitic protozoans. And then when we align these onto each other, and this is aligning the ribosomes. I'm just cutting away the rest of the ribosome and only looking at this helix that's on the surface. We can see that the structure is also very conserved at that very first helix, but there's helices budding off of that. And as you can see, going through more and more complex species, you can see that this helix has more and more helices just budding off of it. And so we believe that this is how the ribosome has, or ribosomal RNA has evolved, is that just helices just budded off from other helices. And so so to wrap that up, the ribosome has a common core, and it's made of RNA and protein. And the RNA is really conserved in structure, but not sequence. And these sites of ribosomal RNA evolution are referred to as expansion segments. And they bud off of the common core of the ribosome, and they do not perturb the common core structure. And this sounds, to make an analogy, it's kind of like a tree. When you look at a tree, you can tell the newest sections of it are the leaves, whereas the oldest section is the trunk. And so we're looking at these structures, and we're seeing these eukaryotic expansion segments. Those are like the leaves and the outermost branches. And when we take away these leaves and take away these branches, we can just go deeper and deeper into the past of the ribosome. And we eventually get to just one helix, and that would just be the trunk of the tree. And this one helix is the peptidyl-transferase center. So it's where peptide bonds are made. And so we can look at how the ribosomal RNA has evolved. Okay, so that's the RNA of the ribosome. Well, what about the protein? Well, the protein is also very similar in some regards. So this is universal protein number 4 of the large subunit, UL4. And so for those of you who are familiar with proteins, or maybe not, I guess, up here we have a globular domain, and that looks a lot like the proteins we see in all of life today. But these ribosomal proteins are very weird looking. They have these really strange tails that just go into the ribosome, and they don't really look like other proteins that we know of. And so a lot of people just don't really look at them, and they throw them out of their analysis and stuff like that. But I'm looking at them because I think they're interesting. And so this is UL4 from a bacterial species, and that's UL4 from an archaean, and that's UL4 from a eukaryote. And then when we look at them within these aligned crystal structures, we see that they line right on top of each other. And they all have this tail that goes deep into the ribosome, and they have this globular domain that hasn't really changed in structure either. But then archaeans and eukaryotes have this little loop here, and then eukaryotes have a little bit more over here, and this large alpha helix that buds off there. And this goes onto the surface of the ribosome, where the ribosome has been evolving. But it all has this common core right here. And this common core goes deep into the ribosome. And it goes right next to the peptidyl-transferase center. The peptidyl-transferase center would be right here. And so it's almost like if you were looking, make like a reaction coordinate of this, that here would be like the oldest peptides, and here would be the newer ones. Like I showed in that last figure, the very center of the ribosome is the oldest parts. It's the oldest ribosomal RNA, whereas the outside is the newest. And when you look at these proteins, it's the same. The proteins, the amino acids and peptides that are closest to the peptidyl-transferase center are the oldest ones, where the ones on the exterior of the ribosome are the newest ones. And so looking at this, we can see how proteins have evolved since the origin of life. So I made this protein folding peak. And so basically what it's showing is that close to the center of the peptidyl-transferase center, we get these unstructured peptides. But then as you move farther and farther out from it, you start saying, oh, okay, well, there's some beta sheets or beta hairpins. And then if you move farther out, then you start seeing beta barrels. And as you go into the surface of the ribosome, then you see these really complex looking proteins that we see in all of cellular life. And so it's a reaction coordinate of how proteins have evolved since the origin of life. And so that's what I have been looking at. And so to conclude these ribosomal protein evolution, you can see that ribosomal proteins also have expansions. And this is very similar to the ribosomal RNA. However, these ribosomal protein structural differences are much more pronounced. We just know a lot more about proteins. Proteins have been studied for a while, whereas RNA, the structures and everything, haven't been very interesting to a lot of people for much time. And these ribosomal proteins, there's a common core to it, and the common core, just like RNA and a protein, is all exactly the same. The sequence might differ, but the structure is the same. And so we believe that ribosomal evolution happens in both RNA and protein, and that RNA and protein have evolved together in a mutualistic fashion. And then so to conclude, I'd just like to show this one slide from a project I'm working on, because we're all astrobiologists here, and it's a little bit talking about metals, and I just maybe want to show some people so that maybe they can give me some input later on. So a lot of these ribosomal proteins are binding zinc. And zinc is very important for a lot of proteins. Looking at any organism that we see around today, 4 to 10% of their proteins need zinc to function. And zinc is found in many of these ribosomal proteins. And these proteins that it's in are usually very simple looking. So here's one of them. And so there's the zinc that's coordinating with 4 cysteines, and it's just made of like 2 beta hairpins. And so when I'm looking at this, it's like, these proteins are usually found pretty deep within the ribosome. And I believe that these could be some very ancient peptides or proteins. And I'm looking at the literature, and there's all sorts of hypotheses of how like the world came about and everything, and what kind of concentrations models were at and everything. And there's some debate about zinc and whatnot. But our lab has also shown in the past that the ribosome needs magnesium cations to stabilize its tertiary interactions between the RNA helices because they're very negative. And there have been studies in our lab and other labs that have shown that RNA doesn't necessarily need magnesium, but it could be using reduced iron. And I found some literature saying that these peptides, these zinc ribbons that these ribosomal proteins have, that they can also bind iron instead of zinc. And so hopefully tomorrow I can look at some posters and maybe some talks, and hopefully someone will be talking about zinc or iron or something like that. But so I guess that will conclude it. There's really not much to put in this presentation summary, but this is our lab, and thank you very much.
All right, we got time for one quick question. I saw Tony back here.
Yeah, so my question, if you can go back 2 slides. Sorry. What are the arrows on the protein fold?
Oh, it's just showing the way you depict a protein is these arrows are just beta sheets, and so that's just how it's structured. And so right in between that, this is a simplistic diagram. You have your amino acids, and they're aligning and hydrogen bonding to each other. And then the arrow is just pointing to oh, okay, so this is the N-terminal, and then this is the C-terminus of the protein. Mm-hmm. So it's just a simple way of drawing a protein structure. Mm-hmm.
All right, thank you very much.
Good, thanks.